Hello everyone. It's a pleasure to share our work biosynthesis gene cluster containing species drive microbial community stability under thermal stress through enhanced ecological cohesion. In this study, we used high-temperature DACO, the starter used in Chinese liquor fermentation, as a model system. Solid-state fermentation heats up rapidly, so we asked whether a few key taxa stabilize communities by BGS-driven secondary metabolism. By integrating metagenomics, iChemp, network simulations, and SynCom co-cultures, we show that temperature reshapes assembly into subset of BGS-containing species stabilizes communities by strengthening interactions and robustness. First, we found that temperature, not fermentation time, significantly drove microbial community changes in DACO. Ordination analyses and ANUSM showed that community composition and alpha diversity differed across temperatures, but not across time. iChemp and mantle tests further revealed stage-specific transitions in assembly mechanisms along the temperature gradient. Bacterial communities were mainly shaped by deterministic processes with process reallocation across temperature ranges, whereas fungal communities were driven more strongly by stochastic processes. We reordered samples by temperature and built 76 molecular ecological networks from adjacent temperature clusters. Richness and total abundance remained stable, while increasing temperature increased compositional differences and shifted node persistence, consistent with heat-driven node loss and network reorganization. iChemp further showed taxon-specific assembly under heat drift increased for bacteria, whereas fungi were dominated by dispersal limitation, with drift also contributing, yielding 27 bacterial and 56 fungal functional groups linked to maintaining abundance and diversity at high temperatures. Eggnog annotation showed metabolism-related functions were prominent, and secondary metabolism genes increased with temperature. Across the 17 dominant genera, we identified 1,413 BASCs, mainly originating from Pasolomyces and Aspergillius. Based on assembly contribution and secondary metabolic potential, we identified 11 key BGS-containing phylogenetic bins. Linear regression indicated that these bins explained 80.38% of the variation in community assembly, far more than other gene cluster groups. Cohesion analyses further showed that BGS-containing bins increased positive cohesion, decreased negative cohesion, and exhibited stronger temperature resilience during heating, linking them to greater community stability. As temperature rose, especially from 51.31 to 61.31 degrees Celsius, co-occurrence distance increased significantly, accompanied by a decline in alpha diversity and an increase in the abundance of BGS-containing bins. This pattern suggests that communities adapt to heat by functional adjustment while maintaining interactions. Importantly, removal simulations showed that 6 fundamental species contributed disproportionately to stability. Removing them markedly reduced both collapse threshold and robustness. In contrast, the absence of BGS-containing bins weakened network topology and interaction density. These results support that these fundamental species act as stability anchors during the high-temperature phase. Per-crust/correlation analyses showed that BGS-driven stability is largely attributable to BGS-containing bins, with 90.3% significantly shaping community BGS content. SynComs further showed that adding the 6 fundamental species increased biomass by at least 50.08%, 3661 degrees Celsius, supporting their role in enhancing heat tolerance, cohesion, and stability under thermal stress. Although not every BGS-containing species showed clear temperature tolerance on its own, SynCom validation demonstrated that inoculation combinations including fundamental species produced significantly higher total biomass across temperatures. Temperature is the key driver of stage-specific dynamics in spontaneous DACO fermentation, disrupting and reconstructing community structure along the heating trajectory. Integrating iChemp with molecular ecological network analyses, we show that BGS-containing taxa strengthen cohesion to stabilize networks under heat stress and identify 6 fundamental species with disproportionate effects on robustness and collapse threshold. Co-culture validation further demonstrates temperature-dependent biomass advantages, supporting BGS-associated fundamental species as practical stability targets in thermally stressed fermentation ecosystems.